Hello everyone, my name is Caitlin Edwards. I'm a student at UW-Casper, and today I'm going to present my research on the disruption of human tubulin citrullination through mutagenesis and its effects on mutant tubulin polymerization and kinesin binding affinity. This is part of a collaborative project with Dr. Sherrington and Dr. Rothfuss at the UW-Laramie. Microtubules are one of the three components of the cytoskeleton and are comprised of heterodimers known as alpha and beta tubulin. These structures are shown to be highly conserved throughout evolution and are involved in numerous cell functions such as mechanical support, intracellular support, and cell division. To perform this wide array of function, tubulin proteins can undergo post-translational modifications. One such post-translational modification is citrullination, which is also known as deamination. This is an arginine-directed post-translational modification that causes a permanent change in the targeted arginines of proteins and is mediated by peptidyl arginine deaminases. More specifically, citrullination causes arginine to undergo a net loss of the positive charge on its side chain, which then also increases the local hydrophobicity of the target protein. Citrullinated proteins are involved in many different cell functions, such as differentiation, nerve growth, embryonic development, cell death, and gene regulation. There's also been a correlation established between pathologic citrullination and diseases such as multiple sclerosis, Alzheimer's, rheumatoid arthritis, and cancer. However, there is currently a limited understanding of the role citrullination plays in microtubule polymerization, kinetics, and kinesin binding affinity. The primary goal of this project is to investigate how citrullination affects tubulin function and structure, specifically as it relates to polymerization kinetics and kinesin binding affinity. Gaining a better understanding of these relationships could provide insight into the mechanisms of diseases related to citrullination and microtubule dysfunction. A brief overview of this project's methods: the TUB3 human tubulin beta gene has specific pre-identified arginine residues that will be replaced with lysine, as lysine will preserve the local charge and prevent the amino acid from undergoing citrullination. This TUB3 gene will be subcloned from a dual expression vector into a smaller cloning plasmid vector in bacteria for easier genetic manipulation. Then site-directed mutagenesis will be performed to generate mutant TUB3 genes. Then these mutant sequences will be cloned back into the dual expression vector, which will then be introduced in insect cells by Dr. Sherrington and Rothfuss in Laramie for co-expression for the different tubulin genes. 3 arginine residues were identified within the TUB3 gene that could be citrullinated. They include R62, R262, and R380. The TUB3 gene was subcloned in a smaller cloning vector, PUC19, to facilitate manipulations and eliminate any risk of introduction of unwanted mutations in non-target sequence. To accomplish this, plasmid mini-preps were done to both PUC19 and the TUB3 vector using the Omega Biotech EZNA plasmid midi kit. Then 2 restriction endonucleases, KPN1 and XMO1, were used to excise the TUB3 gene from its dual expression vector, and the same restriction endonucleases were used on the smaller cloning vector to facilitate insertion of the TUB3 gene. The digestion enzymes resulted in incomplete digestion, so the samples were then purified by electroelution and ethanol precipitation of the DNA to obtain the purified segments before using the second digestion enzyme. Samples were electroeluted and ethanol precipitated again after the second digestion to obtain purified gene and vectors. Once a purified gene and vector set was obtained, the dual expression vector minus the TUB3 gene was kept for future use. The PUC19 and TUB3 gene were then ligated using T4DNA ligase and transformed into chemically competent E. coli DH5 alpha cells following manufacturer's chemical transformation protocol. After the transformation, the clones were initially screened manually using the mini prep kit used to initially prep the plasmids. After prepping the samples, they were checked on an agarose gel against a non-digested PUC prep to see if any of the clones held the PUC19 TUB3 plasmid. When none of the clones from the first round of ligation transformations contained the PUC19 TUB3 plasmid, another ligation and transformation were done. However, this time, to facilitate a faster clone screening process, a blue-white screening using XGAL and IPTG was utilized. The blue and white colonies contain respectively the non-recombinant PUC19 and the recombinant PUC19 with the TUB3 gene. As you can see in the image here, the orange arrows are pointing to some of the white colonies that likely contain the recombinant plasmid that is needed to proceed with the next step. The plasmid will be extracted from these colonies and characterized before proceeding with the mutagenesis of the TUB3 gene. At the time of this presentation, this step represents our current progress, and the rest of the methods presented here will indicate the future steps that will be taken provided a successful clone is obtained. Once a successful clone is obtained, site-directed mutagenesis of the TUB3 gene will be performed using the Q5 site-directed mutagenesis kit following manufacturer's instructions. Through separate codon substitutions, 3 arginine target residues in the tubulin protein will be changed to lysine in all mutant beta tubulin proteins, thus yielding recombinant human beta tubulin that cannot be citrullinated. A total of 3 tubulin codons, each specifying 1 arginine residue in human tubulin beta proteins, will be targeted separately for substitution by lysine codons to eliminate these specific citrullination targets. Separate sets of forward and reverse mutagenic primers were designed using the any base changer software. Shown here is an example of one of the primer designs. All PCR reactions will be performed using the appropriate mutagenic primer sets and the PUC19 tub3 plasmid template DNA. The cycle conditions will vary in the annealing temperature, which is tied to the mutagenic primer sets used. After PCR amplification, phosphorylation, ligation reactions, and cloning of mutagenized plasmids in E. coli DH5 alpha cells, all recombinant bacterial clones obtained will be screened to identify positive mutagenized tubulin clones, those carrying the recombinant plasmid with mutant tubulin sequences. Clones containing the recombinant PUC19 plasmids with mutagenized tubulin genes will be extracted following the same procedure as previously discussed, and the tub3 gene cloned back into its original dual expression vector. The positive recombinant bacterial clones generated will be sent to the University of Wyoming Laramie campus to Dr. Sherrington and Dr. Rothfuss, and they will perform the expression of the mutant tub3 genes in insect cells and all the tubulin kinesin binding and tubulin polymerization assays. Their overall goal of this project is to create mutated human tubulin genes in order to investigate whether citrullination has a role in polymerization kinetics and kinesin binding affinity. It is currently thought that if in vitro citrullinated purified human tubulin affects the polymerization kinetics of and kinesin binding affinity of the tubulins, then there will be a difference noted when comparing citrullinated tubulins to non-citrullinated tubulins. Additionally, this project will determine whether specific arginine residues are required for changes in tubulin polymerization kinetics and kinesin binding affinity. If specific arginine residues regulate tubulin polymerization kinetics and kinesin binding, the modifications of these residues will alter tubulin function. I would like to thank my mentor, Dr. Florence Tula-Finley at UW-Casper, for all her help and guidance with this project, collaborators Dr. Sherrington, Dr. Rothfuss at UW-Laramie, and the Wyoming INBRE Undergraduate Student Fellowship Program for providing me with this opportunity. And thank you for listening.